Hello, good morning, everyone. I hope you had a good uh, engagement week last week and you're a little bit recharged for the second half of the course. We are week 8 this week, and that marks the halfway point of our course. And the test is going to be next Friday, I believe. We have 2 more lectures to go for this unit. And today we will be going through special senses. So let's get started. Of course, as you know, we have 5 general senses: sight, touch, hearing, taste, and smell. We would be spending much of the emphasis on sight as well as hearing, and then we will briefly touch upon the other 3. Okay, but most of the lecture will be on the anatomy of the eyes as well as the anatomy of the ear. Now, if you think back to the nervous system, what we did in lecture, was this 6 or 5? Was that let me just show you the picture to refresh your memory a little bit right here. Okay. So, in order for you to detect changes in the surrounding or even changes that occurs within the body, it all started with some kind of sensory receptor. Okay, so the sensory receptors is the first point for triggering an action potential, for triggering an impulse that would go through your sensory neurons, which then will be processed at the central nervous system, and then the appropriate action would come out through the motor neuron, right? And at the beginning, the sensory receptor is going to be different for different types of senses. So if we are talking about vision, then you would need to have sensory receptors for light. If we are talking about smell and taste, then you would need sensory receptors for detecting various chemicals. And if we are talking about touch and even hearing, actually, you would need sensory receptors that would respond to pressure changes. Okay? So each type of receptor, sensory receptor, is only going to be able to respond to a particular type of trigger. We call these trigger a stimulus. Okay. And so like the photoreceptors in the eyes are only going to be responding to light. It will have nothing to do with pressure, for example. And then the ones that detects temperature will only be triggered by cold or hot, but not by chemicals, right? So they're kind of restricted to the kind of stimulation that it can detect. So once a stimulus corresponding to the receptor is present, then the sensory receptor will release neurotransmitters that will then communicate with the sensory neuron, and then the action potential will be created. And you know the rest of the story. The action potential will be carried to the interneurons within the CNS, and then the corresponding response will come out through the motor neurons, which would then be connected to an effector, right? So let's go through the different types of sensory receptors. First, we have mechanoreceptors. Mechanoreceptors are stimulated by either changes in pressure. So when we talk, we create sound. Sound is basically changes in pressure in the air, essentially. Okay. And when that change in pressure hits the ear, as you will see, that is interpreted as sound. And also, if you take a pen and then you poke yourself on the skin, you can poke it with the sharp end or with the blunt end. It feels different because the pressure is different. So there are mechanoreceptors on the skin as well. So I'll write down for you. Under on skin, I guess, on skin, and also in the inner ear. Okay. The part of the year that you can see is actually just a very small portion. The whole year extends, inside your head. We have the middle ear and the inner ear. So on the inner ear, you have mechanoreceptors. There are also mechanoreceptors that are going to be stimulated by body movement. This is also in the inner ear. also in inner ear. When you rotate the body, when you are falling, or when you are accelerating, like speeding up thing, going on a roller coaster, all those movements that you registers are actually picked up by these mechanoreceptors that are located within your inner ear. Thermoreceptors are pretty self-explanatory. They are stimulated by changes in the external or internal temperature. You can feel temperature of the environment for sure, but you can also feel it from the inside. If you drink something cold, you can feel it go all the way down to your stomach, right? Same thing for drinking something hot. You can feel it going down the esophagus and warms you up, right? So you have thermoreceptors on the inside as well, and even in some internal organs. Okay, so mechanoreceptors, thermoreceptors. The next one is pain receptors. Pain receptors has another name. It's called nociceptors. Nociceptors. Now, depending on the nature of the pain and depending on where it originates from, there are different pain receptors for each of these things. We have somatic nociceptors. Somatic means bodily. So these are things that are kind of coming from the surface of the body, more external things. Right? These are the pains that we are all familiar with. Okay, from things like cuts, bruises. You know, if you bumped yourself, if you burned yourself, right? So all these pain that you register from the surface of the body, from muscles, from skin, those are registered through the somatic nociceptors. Okay, they respond to mechanical. Mechanical means like, like I said, cut, spraying, um, hitting, right? All those things are mechanical. Damage. Thermal is heat. Electrical damage, chemical damage. If you spill acid on yourself, it burns, right? That's a chemical damage. And all these, once again, are registered through the somatic nociceptors. Okay, and then it goes up the spinal cord, and then it goes to the parietal lobe of the brain, right? That's where the pain is registered. Right? The parietal lobe contains, we talk about this in lecture 6, the primary somatosensory cortex, right? So all this information goes to the primary somatosensory cortex located in the parietal lobe. Okay, remember the central sulcus, which is the line that divides the frontal and the parietal lobe, right? So on the parietal lobe, right after the central sulcus, is where you will find the primary somatosensory cortex. So here are some examples of the type of injury that would result in triggering the somatic nociceptors. But that's, of course, not the only kind of pain that we experience. There are pain that originates from the core of the body, more from like internal organs, for example, and those are picked up by visceral nociceptors. And they react in response to excessive stretching of internal organs. What would be an example of an excessive stretching of an internal organs? Can you give me an example? Nothing crazy, just like, you know, common things that could happen to any of us, excessive stretching of an internal organ. Nobody has ever excessively stretched their okay. Spraying, yeah, actually spraying would be somatic, nociceptive, because spraying is more like muscle and ligaments, right? So those things tend to be on the surface, and it would be somatic, nociceptor. Wait a couple more seconds, see if anyone can think of anything. Stretching muscle too much. Again, anything that has to do with muscle is going to be somatic. Okay. Visceral is more of organs on the inside. So an example would be like, if you eat too much, right? Sometimes, I don't know, you go to a buffet or something, you overate, and the stomach is overstretched, and that could cause discomfort or potentially pain, right? That would be, stretching of an internal organs. People might have things like gallstone, right? The gallbladder is attached to the liver, if you don't know, and it makes this thing called bile. And gallstone can form and it can get stuck in the organ, and then it causes it to get stretched out, very painful. If you're not familiar with gallstone, maybe kidney stone, right? Kidney stone, those stones can flow out from the kidney and get stuck in the ureter. That's the tube that drains the urine into the bladder, and that could cause it to stretch excessively, and that could be painful, right? So all those pain would be originating from, again, the inside of the body, the organs, and that's registered through the visceral nociceptors. It can also be caused by oxygen deprivation, right? Such as during a heart attack or chemicals that are released by damaged tissues. Okay, so you might sometimes have like burning sensation originate from an organ as they're being damaged. Various chemicals can come out from damaged tissues, and that would trigger these nociceptors. All right, all the examples are here. Now, sometimes we experience what's called referred pain. Referred pain is a situation where you feel like the pain is coming from one part of the body, but it actually originates from another part of the body. Okay? You might have heard about this. When someone has a heart attack, the pain is not necessarily coming from the heart initially. Okay, there is a kind of like a delocalized type of pain that is originating from the left shoulder area. So that's called refer pain. Okay? You feel the pain from the shoulder, but the source of the pain is actually from the heart. Okay, and these diagrams shows you other other refer pain um, situations. Okay, a pain of the liver and the gallbladder can feel like it's pain from the upper right shoulder, and then we have various organs here that could be hurting, and then it could be projected not from those organs, but like more like a delocalized area in the abdomen. And how this works is this, okay? You don't have to draw this. Okay, so let's say this is the heart, and then this is the shoulder. So this here, the pain from the heart is going to be picked up by visceral. You don't have to write this down. Visceral nociceptors, and then the pain from the shoulder is going to be picked up by somatic nociceptors. And what happens is it turns out the pathway on the way to the brain actually get merged together into the same ascending pathway, same ascending track. Okay? So when it gets to the brain interprets the signal coming from a source that you are more familiar with. Okay. So surface pain, somatic pain is something that we are more accustomed to because growing up we bump ourselves, cuts ourselves and whatnot all the time. And our brain is more accustomed to those kind of signals. And so even when it's coming from a visual source, the brain interpret it as if it's coming from the somatic source. Okay, and that's called referred pain. Does anybody have any question? I guess a more relatable example for referred pain would be like for guys, for example, like if you play any kind of sport, maybe one time the ball hit you down there, okay? And then the pain actually is gonna be in the abdomen, right? That's referred pain, right? Same thing for women. Menstrual pain, even though it originates kind of from the endometrium, from the uterus, but the pain is again delocalized, right? In various abdominal region. Again, the concept is like the source and the perception of where it's coming from, there is a mismatch, and that's what referred pain is. Okay, there is a very rare condition called CIP, congenital insensitivity to pain. It's so rare that there has only been around 20 cases that has been reported. I haven't updated this slide for a couple of years. I think there are probably more cases now, but less than 100 for sure. These are people who do not feel pain, right? They are born with as the name implies, insensitivity to pain. That might initially sound like a good thing, but actually is not. Pain is a way your body tells you something is wrong, that you're hurt and you need to take care of it. So for these kids, they would fall and then they, might break a bone or something, and they would not be aware of it. And then the wound would get, severely worse over time because no treatment was given, right? They might be chewing on their tongue and they would not feel the pain, and then they would have all these damaged tissues, right, all over the body. And there's no fix for it. And because it's so rare, not a lot of research is dedicated to finding the cause and everything. But once someone is diagnosed with this condition, they have to be taught to really take care of themselves and to look for cuts or wounds that they might not be able to feel. And Yeah, it's an interesting condition that you can look up. All right, I digressed. We were talking about different kinds of receptors, right? Mechanoreceptors, thermoreceptors, nociceptors, and now we have chemoreceptors. Chemoreceptors are stimulated by changes in chemical concentration of a particular substance. There are different types of chemoreceptors dedicated to detecting different types of chemicals. So in the taste bud of the tongue, we have receptors for sodium. That would be the sensation of saltiness. We have taste buds for sugary stuff, taste buds for bitter stuff. So all those are examples of chemoreceptors. In the nose, we have receptors for different type of scent molecules. And as we will see, a combination of these sense molecule would activate what we call like a odor signature pattern in the receptors, and that gets sent to the temporal lobe of the brain. More on that later. Other type of chemoreceptors can detect changes in oxygen level, carbon dioxide level. We learned these in lecture 4. We have the carotid body that detects oxygen in the blood. That's located in the carotid artery. And carbon dioxide sensors would be found in the brainstem, for example, in the respiratory center, right? The medulla oblongata, right? That detects carbon dioxide as well as hydrogen ions. We also have photoreceptors. These are only found in the eye, and of course, they are stimulated by light energy. There are two types of photoreceptors, cones and rods. For now, cones are for color vision, and rod is for night vision, night and peripheral vision. Okay, they are not for color, they are just like kind of grayscale. Peripheral vision is like on the side, right? Side view of you. Uh, while we are on this topic, we might as well write down. There are only three colors that we detect. Do you know what the three colors are? Anybody? I know we see all the different colors, but really there are only three kinds of colors. Yeah, red is one of them. Good. Anyone else? If you have one of those old TVs, those big ones, bulky ones, if you wipe the screen with a cloth, then you can see the individual pixels. Yes, very good. Okay, red, green, and blue. Those are the only three colors that we can detect. But it's the combination of these colors that allows us to see all the different colors. For example, red and green together actually makes yellow. Um, we'll talk more about that later on. Okay, let's do some polls here. Go ahead. All right, changes in pressure would be mechanoreceptors. The answer is B, mechanoreceptors. Next one. So remember, this is visceral. Visceral means it is originating from the inside of the body. And so cutting a finger would be surface type of stuff. So that's somatic. Angina is chest pain from the coronary artery blockage. Kidney stone, right? Dislocating shoulder, again, it's more like muscle type of stuff, bone kind of stuff. So that's considered as surface. So the answer would be B and C. All right. All right. Before we move on, there is some important information I need to share with you. We had a department meeting last week, and there has been some changes that we're trying to roll out for all the courses. So starting right now, I guess, our tests will require you to turn on the camera, and it's pretty much what you're doing in math, okay? But now we are doing it for all the subjects. And with the change, notes will no longer be allowed during your anatomy tests. So, please study accordingly. Uh, so it's the lockdown browser plus the camera, and during the test you will not be allowed to use any kind of aid, right? So no phones, no notes. Basically, it's like what you're doing in math. Is there any question about that? I'll make an announcement regarding that on Luminate. But like I said, you've been doing it for math, so it shouldn't be require any extra setup. But I just want you to be aware of that, right? Since it is a shift from what we were doing from the first half. Okay, no questions, then we're gonna move on. Now we're gonna talk about taste. Right? Taste is one of our five senses, and it is registered by the tongue primarily because there are taste buds on the tongue. But taste buds are also found in other places other than the lungs. There are other than the tongue, singular. There are taste buds that are found on the hard palate. So that's the hard part that you feel when you push your tongue up. There are also some in the pharynx and even in the epiglottis. Perhaps that's not difficult to understand. Sometimes if you swallow something bitter, there is this bitter taste that lingers in your throat, right? So that feeling that's like still there in your throat, that's basically the taste buds in the pharynx and perhaps the epiglottis that are continuing to transmit that sensation for you. Okay, so where are the taste buds found? They are actually found along the walls of these cylindrical structures called the uh, papilla for singular, papillae is plural. Let me see if I have a picture for you. There we go. This is a tongue viewed under the microscope, and like if this is the tongue, okay, this is the tongue right here, sticking your tongue out, with all the dot stuff. This would be like mid-sagittal cut. Okay, so like you're cutting the tongue like this, and then you're viewing it. This would be the surface of the tongue. And you can see this whole thing right here, this structure, that's a papilla. Okay, it's 2D right now because you cut it, but in real life, before you cut it, it will be more like a cylinder. Okay, and on the side of the cylinder, you would have these little structures. and those would be the taste buds. Okay, so there is a taste bud. There's another taste bud right here, taste bud right here, taste bud. Right. So when you're eating, the food is gonna be on the surface here, and then some of the chemicals would go in between the papilla, and then depending what's in it, right? Like sodium, glucose, that would stimulate the taste buds, and then an action potential will get sent to the brain, and you would interpret the taste of your food. So we have taste buds for sweet, sour, salty, bitter. Those are the common ones people know. But there is also taste buds for something called umami. Umami is Japanese, means tasty, and it refers to like a savory taste when you eat things like meat, when you eat things like junk food, right? Like the chips and everything, like that savory taste that you get is basically detected by the umami receptor. And like I said, different taste buds is triggered by different stimulus. Glucose will trigger sweetness. H plus, hydrogen ion, will trigger sourness. And then Na plus will trigger saltiness. Umami is actually triggered by the presence of a type of amino acid in your food. And bitter in general is tend to be more basic type of chemicals as opposed to acidic. And all these things will trigger the corresponding taste buds located on various parts of the tongue. Now, you might have seen a picture like this before where the tongue is divided into different regions, right? Like this is sweet and this is like, sour or something like that. But that's actually not quite true, okay? It turns out the taste buds are kind of scattered throughout the entire tongue, even though some region might be more sensitive to a particular taste than another. Fun fact, things tasteless. Things are tasteless in space. Okay, for some reason the taste bud doesn't really work. Maybe without the gravity, the chemicals don't get pulled into the papilla as much. And so for astronauts, they have to bring hot sauce to space to kind of increase their palatability, right, of the food. So where do all the information related to taste goes? We learned about four lobes in the brain. Just to quickly recap for you, we have the frontal lobe here, okay, and then over here is the parietal lobe. Temporal lobe is on the side, and then occipital lobe is at the back. But it turns out if you pry open the lateral fissure, if you peel back the frontal lobe and the temporal lobe, there is actually a fifth lobe that is located behind, and that is called the insula. Okay? The insula, amongst other things, is responsible for processing information related to taste. Okay? So fancy word for taste is gustatory. Gustatory. Okay. Just like sound, we say auditory. Uh, Vision, we say uh, visual, I guess. And for smell is olfactory. So for taste is gustatory. Gustatory. Okay, and that's it. That's it for taste. Let's move on to smell. Smell is registered through these cells called the olfactory cells, and they pick up different chemical related to odor, to scent. And what happens is we don't actually have a single receptor for each of the smell. It's not like you have a receptor for rose and you have a receptor for green tea and receptor for perfume or whatever. Like that would be too many, right? It's not possible to fit all the smell and have a dedicated receptor for it and fit all of those up your nose. So what happens is an odor molecule will activate a cluster of odor receptor, and this cluster of receptors that are being activated creates kind of like a unique barcode that would get interpreted by the temporal lobe in the brain. So let me try to explain this to you. Again, you don't have to copy this down necessarily. Let's say there is a receptor for red odor, okay? Whatever that is, red odor. And then let's say we have a receptor for blue odor, and then we have receptor for pink odor, and then let's say we have one more for these, green odors. So if you smell a rose or something, or some kind of flower, it doesn't matter, and this flower happens to um, trigger the red and the pink, and then red and pink together would be registered as the rose smell. Okay, so this is the rose, put it right here. And what if I have a different thing? What if I have a burger instead? Okay, the smell of a burger. Well, maybe the burger is going to trigger the red one, the blue one, and then the pink one. So when three of them are triggered, that's an other smell that you register, right? So you might have another thing here. I don't know, fish or something. Okay, and for the fish, it's going to trigger uh, just the green. Okay, so if it's green alone, then it's fish. So with four receptors, how many smell can you code? Can you code for? Okay, so the red one, you can either turn it on or off. That's two choices. Same thing for the blue. So each one can be on and off. And if you multiply them together, that's going to be 16 different smell. Right? So of course you have more than four. And let's say you have 10 of these receptors in the nose. That's 2 to the 10 different smell, 1,000 and 24 different smell. Right? So by having a limited number of receptors, you can still recognize a very wide range of scent. Okay, there we go. You don't really have to write down. Like I said, the focus is going to be on eyes and ears. Now, some people are missing certain receptors for certain scent, and we call that specific anosmia. And perhaps the most common one that people might read about online or hear about it is the inability to smell skunk. Right? So skunk, of course, it's not a pleasant animal. It spray out these um, chemicals that contain sulfur in it, and it smells really, really bad. And what happens is people without the receptors for it will not be able to smell it at all. And that, could be a good thing or bad, depending on how you look at it. Okay, gustatory region is found in if you have been paying attention, it's in the insular lobe. Okay, and actually it overlaps with the parietal lobe as well. Okay, I don't think I mentioned it. It's written here, but I didn't say it. Okay, so look at a part in the parietal lobe and part in the insular lobe. All right, so what you need to do when you're studying for the test is you need to know what sense goes to where in the brain. We actually have a summary sheet that we'll fill in later on together to help you kind of map out everything. But you should start thinking about it. Okay, finish. Let's do vision. Okay, vision is next. So eye is of course where you what you use to see things, but the eyes comes with various accessory structures. Okay, so we have eyebrows, for example, at the top. Eyebrows are useful for shielding the eyes from the sun and protects the eye from sweats and debris. Right? If you are sweating a lot from the forehead, you can see the sweat comes down, and when it hits the eyebrow, it goes to the side so it doesn't go into your eye, right, and irritates it. It also creates some kind of reflection to block off the glare from the sun. That's why football players they have these I forgot what they're called, but they put these dark shades under their eyes to reduce the glare from the lights of the stadium. There are eyelids, which when you blink, it helps lubricate the surface of the eyeball. And of course, there are eyelashes that can help block debris and prevents them from entering the eyes as well. Okay, so we're gonna switch to our study guide here, and we will label the eyeball together first, and and then we'll go from there. Okay, so this is an eyeball. Let me try to copy it over. I hope you have it with you. All right, so there we go. Now the eyeball has three layers. Okay, there are three layers that makes up the eye, and we're gonna start with the outermost layer, which is right here. This is the outermost layer of the eye, and it is actually the white of the eye. There's a question, does diagram are included in the quiz or test? Yeah, there could be diagrams. Yeah, but most of the diagrams will be in the lab quizzes, right? Okay, but there could be diagrams in the tests and the quizzes as well. Um, so this here is called the sclera. Okay, I'll write some information for you. Is the outermost layer of the eye, is the white of the eye. When you see the white part of people's eye, that's the sclera. And the sclera allows muscle attachment. Okay, there are quite a bit of muscles attached to the eyeball. Think about all the movements our eyes can make, right? Up, down, left, right, rolling the eyes, right? Those are controlled by muscles. And these muscles are actually controlled by your cranial nerves, which you have 12 pairs of. Now, the sclera is not going to surround the whole eye, because if it did, then the front is going to be blocked, right? And no lights can come in, and that's not going to be very useful. So at the front, the sclera actually becomes transparent. Transparent. And that transparent portion is called the cornea. So the cornea is transparent to let light pass through and is convex in shape. So think of a convenience store. At the corner, there would be a convex mirror, and convex mirror allows like a wide view of the store, right? So similarly, if we have a convex shape cornea, we can collect light from a wider angle. So that is the cornea. All right, that's the first layer. Let's take a look at our second layer. Second layer is right here. It's a little bit harder to color. I don't even know if I can color it. Okay, I'm just gonna draw over it a little bit right here. There's the second layer. Okay, so that second layer is pointed here by D, and it is the choroid. You might have some memory of this word from before when we talked about the choroid plexus. Choroid means it is vascularized, vascularized layer. And so it contains blood vessels and it basically nourish the tissue in the eyeball. Okay? So the sclera is not completely opaque. You can see especially if you didn't sleep well, right? Like you can see the blood vessels in the eye, right? That's basically the blood vessels that are like right beneath the sclera. And then we have a third layer, which is right there. Third layer is right here. And it's the deepest layer of the eyeball. And we call that the retina. Okay, the retina. This is the layer that contains photoreceptors. Right? And remember, there are two types of photoreceptors, cones, which are detecting red, blue, and green. And we also have rods, which are for night vision and peripheral vision. We wrote it down in the PowerPoint slide. All right, what else do we have here? Now, on the retina at the back, so in case you like don't know, right? The light comes in here and then it's gonna basically hit the retina, right? And there is a special spot located on the retina, and this spot is called the fovea. Okay, fovea, or if you read it in textbook, sometimes it may say fovea centralis. We just call it fovea. So fovea contains highest density of cone cells. Right? It creates the sharpest image. Whatever you're focusing on, and in this case, if you're focusing on the writings I created on the screen, then that is going to be projected onto your fovea. Everything else is going to be part of your peripheral vision, and you don't have details about the peripheral vision. I mean, you're aware of what's going on your periphery, but you cannot make out details about those things. I'll explain more when we get to the PowerPoint slides, okay? But you can only focus on one thing at a time, and that thing that you're focusing on is going to be projected onto the fovea. It creates high definition image of whatever you're looking at. And of course, all those information needs to get sent to the brain, and that is gonna be transmitted along the nerve right here at the back. Okay, so this nerve is called the optic nerve. Optic nerve. Send visual, sends visual info to brain. Does anybody remember which part of the brain is responsible for processing visual information? Where does it all go? Okay, if you don't know that, you should review it. It goes to the occipital lobe at the back. Okay, occipital lobe. Now, where the optic nerve is coming out of the eyeball, like this part right here, G, that has no photoreceptors because the space is taken up by the nerve, right? And you don't have place to put the photoreceptors. And without the photoreceptors, you cannot see anything. So that is called a blind spot. Each eyeball has its own blind spot. We will learn how to find it in a bit. Um, Blind spot is also known as the optic disc. Okay, let's move to the front of the eye and see what else are there. Uh, Very important for the eye is this thing right here. Without this, you can't see at all. Okay, so that is the lens. The lens. Okay, so lens is basically going to focus the light rays onto the fovea. Hey, don't draw this. I'm just going to show you, and then I'm going to erase it. You will have light rays coming in, and then the lens will bend it so that it's going to be on the fovea. Same thing. Light rays come in, and then it's going to bend it so that it's going to be projected onto the fovea. And the shape of the lens will actually change depending on whether you're looking at something that's far away or nearby. And for people who wear glasses, uh, it's because the shape of the lens might be changed or the length of the eyeball has been changed. So we will talk about those things as well in the PowerPoint slides. Okay, so that's the lens. Um, I should mention it. It is a converging lens that focuses light onto fovea. A converging lens is a lens that looks like this. Right? It causes beams to come together. Think of a magnifying glass. That's a converging lens. Right? The opposite of a converging lens is a diverging lens. So, like a lighthouse at the shore, they want to send the beams out spread them as far as possible for boats to see them. Right? That would be a diverging as opposed to a converging lens. Okay, so inside the eye, we have converging lens. Now, the lens is held in place by these ligaments. Okay, these are called suspensory. Suspensory ligament. Okay, holds the lens in place. Now, I'm just going to switch to, uh, give me a moment. I'm just going to switch to the 3D model to show you. This is on Luminate, okay? Under the 3D model folder. So again, it's the cross section of the eye. This would be like looking at it from the front. And you can see this is the lens, and the lens are held in place by these ligaments. If I turn the eye a little bit, I guess, then you can see it's like all the way around, right? The lens is like a full circular structure, if you would, and all around it, you would have these ligaments that are holding it in place. Okay. So one end of the ligament is connected to the lens. The other side of the ligament is connected to these muscles. Okay. They call it by a different name here. Uh, it's the ciliary muscle. Okay, we're gonna label it together. While we are in this 3D model, let's just go through a few other things. You can see the sclera, which is the white of the eye. But as you go to the front, the sclera becomes the cornea, right? Becomes the cornea. It's transparent, see-through. Underneath the sclera, we have the choroid and the retina. Fovea is right here. I don't know if you guys go to the eye doctor regularly, but if you do, they sometimes will take a picture of your eye, right? And next time if you do that, you can ask them to show you the picture. And on the picture, you will be able to see your own fovea as well as the optic disc, which is the blind spot, right? Okay, back to here. The other end of the suspensory ligaments, as I was saying, would be connected to a different structure, a muscle right here. You can't really see the whole thing because it's a cross section. Remember, it's like, going all the way around. And that is called the ciliary muscle. Do we have a label for that? Actually, I can't really see um, where the things are pointing at. Okay, I think maybe okay, I see it wrong. I saw it wrong. The suspensory ligament is actually N. That's what N is pointing at right there. So I'm gonna move this over here. Sorry about that. I couldn't see the lines very well. So that's the suspensory ligaments. Try it one more time. Guess the key point is not like don't memorize the letters, but like remember what they are pointing at right here. That's the suspensory ligaments right there. Holds the lens in place. And then B is actually pointing at the muscles right here. And that would be the ciliary muscle. So more on that later in the PowerPoint, but the ciliary muscles will control the tightness, the tension, right, of the suspensory ligaments. Will we get blank? You mean like fill in the blanks for the test? Is that what you mean? Renew? Or like do you mean like blanks on a diagram like this? You might. Yeah, you might. Yeah. All right. When we go through the PowerPoint slides, I will tell you how the muscles works with the ligaments and in changing the shape of the lens as we shift focus from far away to nearby. All right. Another thing here. Uh, Actually, we'll get to A in a bit. Um, to print and learn the label. Yeah, this is in the oh, you mean like a blank diagram for you to learn. Yeah, yeah. This is in the study guide. Right? All the diagrams I'm doing with you is on the study guide, right? So like that's why I encourage you guys to print the study guide out. You see the ear and the brain and all those things. Okay. All right. Like I don't know how many of you here actually have the printouts, okay? It's pretty ineffective if you don't have the printout and you're just watching me do the coloring, right? Like it doesn't make sense. So you need to print these things out. Okay. Oh, thank you. Okay, some of you are trying to encourage me, telling me that, oh, you have it. That's great. Okay. Now I'm gonna switch to this diagram here for a second. the eyeball is actually separated into front and back by the lens right here. Okay, so this is the lens. This is lens. And the front here is called the anterior chamber. And obviously, if this is anterior chamber, this would be the posterior chamber. Okay, anterior and posterior chamber. Now, they are not empty because if they were empty, then the eyeball would just collapse, right? Would just like close on its own and, you know, flatten out, and we don't want that. So the chambers are filled with kind of like a gel-like substance, okay? So over here at the back, you can see some of that gel-like substance. It's, colorless, transparent. We we don't see them. We allow the lights to pass through them. So that's the gel right here. And the gel fills up the whole chamber, okay? They just draw a little bit of it instead of filling up everything because they want to show you the fovea and everything, right? So this stuff, this gel-like stuff is called vitreous humor. Okay, vitreous humor. And the function of vitreous humor is basically to reinforce the shape of the eyeball. Okay? It props the eyeball up so it doesn't collapse. But we don't want the pressure to be too great. If the pressure is too high, then it actually will compress the optic nerve and it could lead to a condition called glaucoma. You might have heard of it. If not, don't worry, we'll learn about it in the PowerPoint slide. Excuse me. Now at the front here, oh, sorry, another label here. P is just pointing once again at the posterior chamber. Now at the front here, we have another liquid that fills up the space between the lens and the cornea, fills up the anterior chamber. And this is called the aqueous humor, the funny water, aqueous humor. And what it does is it actually nourishes the lens and cornea. You cannot have blood vessels going through the lens and the cornea. If you have blood vessels on these things, then we would see it in our vision, field of vision, right? We will be able to see those blood vessels because as light pass through them, the vessels will be in the way. So we cannot have blood vessels there. And yet these are structures that are alive and they require nourishment. And that's what the aqueous humor is for. Okay? It's there to help um, constantly provide nutrients and wash off metabolic wastes. Your eyeball keeps on producing aqueous humors. Some of it will go to the back and become the vitreous humor, but much of it will be drained at these little drainage points here and there. The drainage point, they are called sclera. Sclera is just the adjective for sclera. And you might remember this next part, okay? It's called a venous sinus. Okay? We previously learned about the dural venous sinus in the brain for draining CSF. So there are other types of venous sinuses for draining other fluids. And in this case, it's found in the sclera. And this one drains excess aqueous, aqueous humor. Okay. I think we cover Most of the stuff. Oh, there's M right here. M is the color part of the eye. Different people have different eye colors, right? Brown eyes, blue eyes. It's related to melanin, the same pigment that gives us our skin tone and hair color and all that. So that is called the iris. Iris. It's the color part of the eye. But it does more than that. It's not just to make our eyes look pretty. There is a hole in the middle. The hole in the middle is the pupil, and that's where light goes in, right? So the size of the pupil is important. If it's bright, you want the pupil to be smaller. If it's dark, you want it to be bigger. So the iris helps control the size of the pupil. Control size of pupil. Right there. And that's it. Okay, don't worry about J. Yeah, not important. All right, before we return to the slides, does anybody have any question? Okay, great. Let's go through a few more slides. Can I explain what M? What's M? Let me see. M. Oh, iris. Yeah, iris is the colored part of the eye. Um, Here, just hold on to that thought. Maybe it's better when I show you the PowerPoint slides, okay? Because then you can see it from a different perspective. And if after I explain it and it still doesn't make sense, just ask me a question again. Okay, so this is just another eyeball looking the other way. It's a good idea to try to go through the labels on your own again and match them up. It's going to help with your understanding. Like I said, sometimes they instead of calling it just the fovea, they say fovea centralis. Doesn't really matter. Same thing for our purpose. All right. So starting with the outermost layer one more time, and it's the white of the eye. I mean, it could turn other color. It could turn reddish, pinkish when you have an infection, pink eye. Um, and actually, hold on.